Hi students, this is one of the most important video in the biomolecules. Why? Because in the total biomolecules chapter in the 12th class, 15 structures are given in one page, that is in the biomolecules. All these 15 structures are coming in this. And applied into the structures and explanation, it is understanding is also difficult, but I will make it easy to you. And questions also used to come from this in the NEET. But direct questions only once he asked about the listening. But the thing is, he is asking wrong statement, right, about wrong one, and this thing matching like that he is giving. If I discuss those things, which I didn't discuss up to now, also will come because of the future topic that is. So I will, at the end of the topic, I will discuss those things. All this is biomolecular topic. Now, anyhow, whenever it comes, I will tell you in which way it is important. Now, amino acids, he has not given heading, and lipids he has not given, nucleotides also he has not given, but he explained in the depth by giving structures. But I given for your comfort, convenient. First, we will see the amino acids. Amino acids are the organic compounds. Organic are the organic compounds. They are formed by the substitute of hydrogen from the methane. What is methane? Methane is this one. Methane means here one carbon, 4 valency will be there. In the 4 valency will be occupied by the hydrogen, by the covalent bonds, and methane will form. So all the 4 carbon valency carbons are occupied by some other thing. Then this carbon is called as alpha carbon because he mentioned alpha carbon in the textbook for that only. Whatever the textbook he has given, every word I am going to explain. So in the alpha carbon, these hydrogens, whatever are there in the valency position, out of 4, 3 will be replaced and will be filled by new groups to form a amino acid. Understand now? Out of 4 hydrogens present in the methane, 3 will be replaced by the different groups and it will form the amino acid. Then we used to call the amino acid as alpha amino acid. For example, now what are the 3 groups? One is the carboxyl group, COOH, carboxyl group. Second is the that is the amino group. Third one is the R group. So these things, how it is. Now whenever we are seeing here, that I shown clearly also. This is the thing. And the substitute, what that the substituting carboxyl group will be removed by came here by removing one hydrogen, and the amino group will come someplace anywhere here by removing one more hydrogen, and R group, which is also called as functional group, will replace one more hydrogen. One hydrogen only will be same place it used to present. Because any angle he can ask the question. Remember it. For that only very simply I am telling you. So all these three places are occupied by different groups by replacing hydrogens. And this all are present on the single carbon, so it is called as alpha amino acid. Understand now? So in this way, amino acid will be formed. So amino acids, whenever it is forming, R group is the most important. These are the amine group, carboxylic group are common for any type of amino acid. But R group used to change, functional group we used to call. How many amino acids are there? Many, up to 100, more than 100 also present. But only 20 types of the amino acids are participating in the formation of the proteins. Protein forming amino acids we used to call. So how many are there protein forming amino acids? 20 are there. These 20 how they will differ one to another? By the difference showing in the R group. R group is a functional group which is to differ from amino acid to another amino acid. And carboxyl group of amino group are not differing from amino acid to another amino acid. Understand now? So R group, whenever we are seeing here, now you see, for that one he has given very simple. This given in the textbook. I brought it separately by editing. Now you see, this is the one amino acid carboxyl group, amine group, and hydrogen. One is left because it is not replaced, and other place also one hydrogen came. Means here the R groups. These are the R groups. The R group is simply hydrogen only. Then it is called. This is also R. R group is hydrogen is present. What it is? It is amino acid name. That is, we used to call it a glycine. Glycine is the simplest amino acid. Remember it, all these are the lipids. Simplest, because R group is represented by only one hydrogen. Or you can tell one thing also, that is, two hydrogens are present on the alpha carbon, and one place is the amino group, one place is the carboxyl group. Then what we can call it? We can call it as glycine. That is the thing. He can ask in any angle. Concept you should know. So R group is represented by the only hydrogen. Then it is glycine. And its molecular weight is, for your reference I am telling, 74. And all the 20 amino acids we see up to 204 it is. Highest molecular weight is the, means that is the, we used to call it as a the molecular weight. So molecular weight up to 204, that is the, that whenever it comes I will tell that also. So why I am telling minimum is the 74, maximum is the 204. In between where all the things used to present means in the R group. R group is working as a tail, or it may be worked as a single hydrogen also. For simple things he has given here. Only one hydrogen is present in the R group, then it is glycine. And methyl group CH3 is present, then it will become alanine. And hydroxymethyl group is present, then it is called as serine. These three structures he has given. All other structures are the most complicated. Means the R group used to have long chain, or it may be having some circular, means aromatic compounds also it will be there. Aromatic means structural, like benzene. So all these things will come in here. So glycine is to have the hydrogen only in the R group, and alanine, methyl is to present, and serine it is to. And need to persons who are going to need, he will give the structure, and he won't give the name. He will give the genetic code. That is the need bit. You should know what is this one, and it's a genetic code you should identify, and multiple choice or he may give, or statement he may give. Understand the NEET means how many, how it will be converted. Such type of things only your brain has to work. Then only that NEET examination it is. Understand? But first time who are listening, okay, this will come. I will do that video also. There also I will discuss these things again. Remember it. So here, three amino acids, it is formed like this. Now, one more thing is, and here carboxyl group whatever is there, that represents the acidic nature. This is also important. And whatever the amine group is here, it represents the basic nature. And here both are the, though it is neutral up to here, but R group, I already told, a lot of so many molecules is to present. If they also used to have carboxyl groups, amine groups also in that one. If carboxyl groups are more in the R group, then it will work as acid. If the amine groups are more, in the R group, it will work as the basic amino acid. This is important for the need and given in the textbook also. Carboxyl groups are more. Generally, they want more here, but in the R group, there it is more. Then that will be the acidic, and amine groups are more. Then it will be the base group. Generally, it is to present majority of the amino acids are neutral. And acidic group, if it is appearing, those are called, that only has given one paragraph. This is the textbook only. Now you see, the chemical and physical properties of amino acids are essential. Physical and chemical properties of the essential. That is the carboxyl group and reflecting the groups. All the group I told, no? Physical chemical characters, how it is. Carboxyl group is the acidic, amine group is the base. R group both used to have, depend upon the number, it is to show the either acidic or base or neutral. That only you want to tell. Now you see here, based on the number of amino and carboxyl groups, they are acidic, same thing, but confusingly you wrote. Not on the alpha carbon, in the R group, remember it. But he not clearly mentioned R group. That is, if there are the acidic, he has given one example, that is glutamic acid, and basic lysine, and neutral is the valine. And now here I want to tell one more thing. More than out of 20, 15 or above are the neutral. Neutral also so many are there. Means that is it is having the non-polar, polar, all the different things are there, not necessary to you. So neutral it is. Means charge is neutral. So neutral are more, but it is given only one example. That is the valine. And persons who are going to the NEET examination next month, please see these whatever amino acids is given, they genetic codes also. But who are following my videos, when I come there I will ask you to practice. But already who completed and going to NEET exam, it is important whatever he has given the amino acid must be read in the form of the genetic code also, remember it. And one more thing, aromatic. Some are similarly, there are aromatic amino acids. Aromatic means what? Cyclic, benzene, cycle. And sugars, majority are the sugars also. That is glucose, fructose, all these are the cyclic. Such type of the cyclic arrangement things are there, where they won't be in this one. They will present in the R group. R group is very big one. In that R group, cyclic molecules are there, structures are there. Then it is called as aromatic. That is called as aromatic amino acids. What are they? Aromatic amino acids which are cyclic: tyrosine, phenylalanine, tryptophan. This tryptophan is the 204 molecular weight, highest molecular weight. So cyclic compounds are present in the R group, in the amino acids formation, alpha amino acid formation. Then it is called as aromatic. All these are the neat bits only. And one more thing, jetter. Majority people are getting doubt. What is jetter? Jetter means here, already I told, carboxyl group acidic and amino group is the base. When both are present, acidic and base are present on the same, and this R group is also almost same, means charges and all the things, polar, non-polar, whatever it may be, they will work As acid in the acid media, they will work as acid. Same amino acid in the acid media, it will work as a acid. In the neutral media, it will work as neutral, and in the base media, it will work as base. That is called, means like this whatever it is to change its structures according to the pH. According to pH be less than 7 is acid, above 7 is the base, 7 is the neutral. If the solution, whatever if we are keeping that amino acid, the solution pH used to change its structure, then it is called that amino acid is called a zwitterionic. Zwitterionic. That only he is telling. Here see, a particular property of amino acid is ionizable natural nature of amine carboxyl groups. Hence, the solutions differs the pH. The structure of amino acid changes. Understand now? That is called a jetter. How it will? Now you see here. He has given one example in the textbook only. That center B, it is the neutral. I wrote here, note down in the textbook also, our book also, 7 pH. There it is neutral, even though it is having positive charge and negative charge. When it will become neutral? When R group is neutral, remember it. He can ask. If R group is charges are there, then it is no chance of this thing. Now, if it is below, means acidic nature, below 7 pH, what happens? The carbonyl group, it will be COO, carboxyl group will lose already one hydrogen and proton, means it may be. And this water amine group NH2, it become NH3 by taking one more hydrogen, it become NH3 positive charge, and CO is the negative charge. Both the charges are present, so it is the neutral one at 7 pH. The same amino acid, when you shifted one medium, by giving this one, which medium it is acidic. Why? Because it become neutral. COOH is neutral. Hydrogen came and joined one proton, and this remain like that only. NH3 positive charge, so it exhibits positive charge. Free protons are present where in the acid hydrogen proton. That is the thing. So it is acid media below 7 pH. Now above, whenever the same amino acid shifted, the jitter nature, how it will work? NH3 will become NH2, which is become neutral because it has lost the one proton. And this will remain like that only. Carboxyl group will be remain like COO, means minus will be there. One hydrogen it lost. So negative charge will be exhibited, and this is the positive charge is a neutral thing, and it is a base. So it is acidic media, base media, and it is neutral. And one more is there. This is the polar, non-polar. Polar also there. Polar means it is having the charges. Non-polar is also there. It is not necessary to you because you will get confusion more. Now we'll see. So up to amino acids clear now? Everything is clear. If you want, listen once more. This whatever I thought in this video, and note down the things in our textbook. And lipids. This is also one of the confusing topic in the NCERT book. And diagrams also he has given, some unnecessary also given. That also I am going to show you. Lipid. Amino acids are important. Similarly, lipids also very, very important. Mainly nerve cells working and sensitive cells will work with lipids only. Lipids means fats. Fats is not like lipids are most important for the nerve system. In the last sentence he given, complex lipids are present in the nerve cells. Important need bit it is. Now you see, lipids are, most of the lipids are not soluble in the water. This is one bit. For that only it used to appear as the macromolecules. Means return date it used to come. That will come later on. That I will discuss. And next, they are present in the fatty acids, glycerol, etc. And means lipids are classified in different ways and different standard people only. One of the way, lipids are whatever they're classified. The first one they are given as the simple fatty acids and glycerol and phosphate containing phospholipids. They are present in all the classifications. So here we will consider it as fatty acids, simple fatty acids. Remember it. So simple fatty acids. Now simple fatty acids has R group attached to the carboxyl group. In the amino acids, carbon it is attaching to the carboxyl group, amine group, R group. So many all these things are there. But here only one very easy to remember the fatty acid. Only one carboxyl group attaches to the R group. That's all. No other groups. That is like this you can show. This is the COOH carboxyl group and R group. So R group it is to attach. Which one? That is the carboxyl group. R group is to present generally in the fatty acid 3 types. That also given in the textbook. But you write it like this, it will be easy to understand. In the book, whatever has reading and confusing. That's all. Write it like this. So R group may be methyl CH3 and forms the fatty acid, or ethyl group C2H5, or higher number of that is the methyl groups, that is from 1 to 19 carbons. This is most important. So highest carbons in the fatty acids can be seen in when it is having the CH2 group chain. But limit is there. Limit is 19 only. It won't cross. The highest carbons can be seen in the simple fatty acid is 19. Okay. So simple fatty acids won't cross 19. But if you include where it is, they are present to the carboxyl group. Here one more carbon is there. That carbon also if you add, it will become 20. That only has given. For example, palm tonic acid 16 carbon. Out of 16 carbon, 15 are from the fatty acid, and that included carboxyl group. One more carbon, it becomes 16. Most important. He will give the same bit. Palm tonic acid, fatty acid contains of how many carbons? Immediately the person will put 16. No. He given fatty acid. Palm tonic acid formation participating fatty acid, yes. 15 only. But totally 16. These are the neat questions. But he is not going in that way. Otherwise, if a person like me gives me answer, that botany, what is botany, you will come to know. Now, please listen properly. It is not a confusing. If you listen and concentrate properly, you will get 100% good marks in the botany. Please, okay? Please accept this word. And palm tonic acid, 16 carbons, including carboxyl group, excluding that one, 15 only. And arachidonic acid has 20 carbons. In that one, it is having the long chain of the containing of the fatty acid, 19 carbons, plus 1 in the carboxyl group. It becomes 20. So, 20 including carboxyl group. And now one more thing is, fatty acids, we used to call the fatty acids saturated fatty acids, unsaturated. Saturated means they are not having the double bonds in between the carbons, because what it is, it has given ethyl group or methyl group something, or the CH2 group chain. In between them, there won't be any double bond in between the carbons. Then it is called as unsaturated. If the double bonds are present one or more than one in that R group, then it is called as unsaturated. Unsaturated fatty acids participates in the formation of the oils. Saturated fatty acids participate in the formation of the fats. This is also need bit. Remember it, write it somewhere, underline it, or write it like this by seeing later on this video. Once more, you write it as a notes. Then you won't get any confusion. So, fatty acids are saturated without double bonds between two carbons. Unsaturated, they will have the double bonds in between the carbons in the R group. One may be there, many may be there, then it will become unsaturated. And next he is telling one more important thing, that is the glycerol. Already in the starting I just told, according to the classification, simple fatty acids and simple glycerol and some other things also there in different classifications of the lipids. Now here he discussed two only, that is glycerol and fatty acids, simple one. Now whenever we are seeing here the simple lipid glycerol, glycerol is also called as trihydroxypropane. Remember it, this is need bit. Trihydroxypropane, glycerol. Because three hydroxyl groups are present in this one. This is the thing. Three hydroxyl groups. And it is a present glycerol. Trihydroxypropane we used to call. And these trihydroxypropane, glycerol, hydroxyl groups will be useful for attachment of the fatty acids or some other things. How many places it can be able to attach? He can ask three places only, because three hydroxyl groups are there. Only three places only it can attach. If it is attaching fatty acid at one place only, then it is called monoglyceride. If it is attaching two places, diglycerides. If it is attaching three places, triglycerides. So monoglyceride, diglyceride and triglyceride, how it came, depend upon the how many hydroxyl groups it is attaching to the glycerol. Understand now? So the glycerol, three places it can attach. That is going triglyceride, R1, R2, R3 are the R groups. And the fatty acid groups is there. They are coming and attaching. But you see, this is the same. This hydroxyl group is attached. Hydroxyl group, whenever it is attaching to the fatty acid, that is esterified, ester bond. This is also important. Ester bond will be formed when the fatty acid is attaching to the glycerol. Bond name is the ester bond, that is esterified given. So triglyceride, how many ester bonds are present? He can ask three. Three ester bonds are there. And next, whenever we are seeing here, it is not mentioned, but palm tonic acid formula he has given. Just simply by heart it, and now and then you see. If whenever he given anything in the NCERT book, chances are there to ask. He is having the right to ask. So this is like that. And already I told the lipid, whatever the glycerol, whatever the triglyceride, other thing, phosphate also can be attached to that. Phosphorylation can be taken place with glycerol. So here, one of the hydroxyl group will be attached with the phosphate. So in the phosphate is whenever it is attaching, how many chances are there to attach fatty acid two places only. One place is attached to the phosphate. So phospholipid, phospholipid, where it came? Already it came in the phospholipids bilayer is present in the plasma membrane. Those phospholipids used to have the phosphate is the polar head and tails of the fatty acids, all the things you studied there. So here these are the fatty acids, the two, and this is the phosphate, and this is the special phosphate that is a phospholipid lectin. Most of the living organisms used to have the lectin as the plasma membrane. The biphospholipids are there, no? That biphospholipids, most of the things used to have the lectin. This is also important. And this why he has given it, tell me. This one more, he is not a madman. All these are participating with our group or some extent or the glycerol, they are participating. But what is this? This is the thing which is not following these structures, but it is a lipid. Cholesterol is a lipid, but it is not following either fatty acid structure or glycerol structure. So, but it is all the functions are like fatty acid, steroid it is. For that showing, he has given 3 hexagonal rings and 1 pentagonal ring, cholesterol. He can give this one also in the NEET. What is the name of this? Means it is the cholesterol. Cholesterol is nothing. Only it is the one of the lipid which is not following the formation of the fatty acid or glycerides formation. But it also used to have the limited number of the carbons like that. And means if jointing means 27 is there, he has given the formula whenever we are seeing. It is different. But the thing is, one thing you remember, it is a lipid, but not following the structures of these. That's all. That is enough to you. Now, you see here. Now, one more thing. Already I discussed this one. These glyceride forms oils. Oils, what I told, unsaturated works as the oil. Saturated fatty acid is present in the glycerol and forms the monoglyceride, diglyceride, triglyceride. They will become unsaturated, become oils. Saturated will become fats. And fats and oil difference is there, yes. Fats means it will be in the solid condition at the room temperature. Its melting point is very high, but oil's melting point is low. That also he has given in your book. So, and one more thing I am telling. Generally, animals are used to store in the form of the fats. Plants used to store in the form of oils. That is also important one, but not out of it is. So that only has given solid at room temperature. If it is, that is the saturated fatty acids. Means what? It is not having the double bond, carbon between carbon. Oils used to have. That only he has given. And one more thing he has given. Phospholipids used to work in the formation of the cell membrane. That already I discussed. Plasma membrane means biphospholipid layer. There the structure is a glycerol with fatty acids and phosphate. These all are the neat bits. To remember these things only, you will get, but you have to see it 2 times, 3 times at a time, and every 1 week you have to see once more. You get even this chapter, if you follow this one, you will get how many marks you are getting. Yesterday also I told. Last cancelled examination 5 bits given. Generally 4 to 6 bits are coming from this chapter. So that much important topic. Now we'll see most complex lipids are present in the neural cells. He has given in the final sentence. So complex, highly complex lipids are present in neural cells. That is also a little bit. Now we'll see about the kinds of nucleotides. This only generally people used to get some confusion. What is this one? Why he has given what? It is very simple to understand and to remember. But second year people, when they studied this molecular basis inheritance, they will feel easy. But first year people, means 11th class people, whenever they are studying, they will get lot of confusion. For that also I am telling you, Kitty, I am going to teach you in the easy way. That is, nucleotide consists of the 3 components: phosphate, sugar, nitrogen base. 3 components used to present in the nucleotide. Nucleotide, you know, from the 8th class, 9th class, 10th class it is coming. Nucleotides are the monomers to form the nucleic acid, DNA or RNA, whatever it may be. So here 3 components are there. In that one, whenever we are saying the nitrogen bases are 5 types of the nitrogen bases are there, but only 4 used to present. Either in the DNA 4 only is present and RNA 4 only used to present, but there are 5 nitrogen bases are there. Why it is it coming, then I will teach there. Now you remember how to answer at present. Now you see, here he has given purine. Adenine is the purine. Purine and pyrimidines. Nitrogen bases 4 are there. One in the 1, 3 are there. 1, 2 are there. Purines used to have 2 nitrogen bases. They are adenine and guanine. I wrote here. Note down in the text also. And pyrimidine used to have 3: cytosine, thymine, uracil. Means either uracil or thymine, only one used to present in the DNA and RNA. RNA, uracil used to present in the DNA, only thymine used to present. Means either cytosine, thymine are present in the DNA or cytosine and uracil are present in the RNA. That's all. That will come later on. Now I am telling purines and pyrimidines. Purines are the 2, guanine and adenine, and uracil, it is the cytosine and this thing. Now here adenine structure, nitrogen base structure he has given. Why he has given the 2 structures? Any difference is there? It is important little bit. Double ring structure is there, it is purine. Single ring structure is there, it is pyrimidine. But the only thing is small changes will be there. Purine and this adenine and guanine, that is the oxygen will be more in this one. And here some amino group may be attached. Like that, very small differences will be there. But basically there are the like this. Double ring is there, purine, adenine and guanine, double ring. And cytosine, thymine and uracil, they used to have the single ring. Understand now? Now this thing, I already told, that is the nucleotide will be formed by the 3 components. That is the nitrogen base and pentose sugar. In the pentose sugar, and it used to have the one more phosphate. So nitrogen base and pentose sugar are attached, it will become one unit. And pentose sugar, nitrogen base and phosphate is attached, it will become other unit. Understanding now? How? For example, here adenine and this is what is the pentose sugar. That is called adenosine. Adenosine. Adenine is the nitrogen base. With pentose sugar, it is having only two components are there, sugar and adenine nitrogen base. That is called as adenosine. Guanine is there, guanosine. It is called as a guanosine. And same thing will be applied, that is the pyrimidine uracil is there with pentose sugar, then it is called uridine, not uracil. And cytosine and pentose sugar, it is called cytidine. Thymine is present with pentose sugar, thymidine. Why? What I feel, this is my opinion. Cytosine is there, no? Cytosine and again nucleoside if you added, cytosine sign you have to say. It is not good. For that only they might have done. Dine they added. And you have to know the difference. Purine and pyrimidine. Pyrimidine is coming there. That is they added. So that is cytosine. That is the adenosine and guanosine. And cytidine, thymidine, uridine. When it is attached, that uracil or cytosine or thymine are attached to the pentose sugar, it is called as cytosine is called cytidine, thymine is adding thymidine, and uracil add uridine. Now one more thing comes. This up to here only two parts are attached. Now nucleotide formation, whenever we are seeing here, this nucleoside, if we attach it to the phosphate, then it will become nucleotide. But the nucleotide is called in a different way, because guanosine and the adenine. Adenine is attached, adenine containing nucleoside attached to the phosphate, then is got adenylic acid. Acid means phosphoric acid it is. For that one they might have kept. Adenylic acid means adenosine plus phosphate, it will become adenylic acid. Guanosine plus phosphate, that is the guanolic acid. Cytidine plus phosphate, cytidylic acid, and thymidine plus phosphate, thymidilic acid, and uridyl and phosphate, uridylic acid. Means by seeing this one, if you wrote this thing, all the things will be easy. In this one, that much only he has given. And don't get any confusion, all the diagrams I shown here. Whatever he has given 15 diagrams in one page. By seeing the page only, some people used to feel afraid. What are these things, all the things. Now it become easier or not? If you want to see the video once more. Note down all the things in the textbook or prepare notes. And you will get 100%, you will give the answers for any type of the question comes from this one. In the tomorrow lesson, next it will be continued. That is the secondary metabolites and other things. I will continue. Tomorrow also I will give one more video. Because already yesterday I given one word to the audience. That is, I am going to complete this biomolecules by the month ending, because it will give some help to the students who are going to attend on the 21st of the June exam. Because any type of the even very merit student also used to have some small doubts in the mainly biomolecules. If we listen this one, everything will be clear. Very clearly I am explaining. If you like this video, please give the like mark, because it will reach to the many people. That whatever the YouTube, it is to show the likings and views, this only, not for me. It must be useful for many students. Means you have to, if you like, give it the like mark. And if you want to see all the my topic, whatever the videos, subscribe the channel. Why? Because immediately after uploading one video, it will come to your mobile immediately. This has came whether to follow or not. Otherwise, you have to search and all the things. Is it necessary? Not necessary. Just subscribe it if you like my way of teaching and the presentation and the thing. Okay, thank you.